Our paper deals with a medically very important issue, namely the gender specificity of disease disposition and drug metabolism. A very serendipitous finding led us to study the aspect that is in the paper published in the Journal of Hepatology. While we were doing inducing experiments on UDP glucuronosyltransferases in an animal model, we observed that induction by coffee in male animals was significantly higher than in female animals and was tissue-specific, namely induction in liver and stomach was observable both in females and males. However, induction in colon and jejunum was not and was specifically only observed in male individuals. This is a very interesting observation and requires more study, and this is why we performed the present study. UDP glucosyltransferases are an enzyme family which is very important for drug metabolism on the one hand, but also for the defense against antioxidative mechanisms and for the detoxification of a number of compounds capable of inducing inflammation and carcinogenesis. Therefore, the tight control of this multiple enzyme encompassing family is something that guarantees the stability of metabolism and therefore the stability of cells, preventing cyto and genotoxicity. In the present study, we were able to show that there is obviously a difference between males and females, and this goes along very well with the observation that certain diseases such as viral hepatitis, cancer disposition, and inflammatory diseases such as autoimmune diseases have a very clear gender dependency. So in the first line of experiments, we sought to figure out whether this is reproducible in cell culture models. To this end, we used three different cell lines. Two cell lines deriving from liver and esophagus, namely Ki-70 cells and HepG2 cells, which originate from male individuals. As a control, we used SW403 cells, which are derived from a female person. This way, it was possible to show that indeed induction of UDP glucuronosyltransferase genes is more effective in the male-derived cells as compared to the female-derived cells. And we then wondered what the reason for this might be. And obviously, if there's a gender specificity, one would look towards gender-specific genes such as estrogen receptors, for example. So we were looking at estrogen receptor alpha 1 and found that the expression of estrogen receptor alpha 1 is much higher in the female-derived cell line as compared to the male-derived cell line. In order to counteract this effect, we then treated the cells with tamoxifen. And interestingly, it was possible to restore inducibility in the cell lines by pretreatment with tamoxifen. In addition, we performed co-transfection experiments in which estrogen receptor alpha 1 was co-transfected into the cells in order to show that the presence of estrogen receptor alpha 1 reduced transcriptional activation. This was indeed the case. And even when cells are induced with dioxin, which is a classical inducer of UGT1A genes, via XRE, xenobiotic response element mediated mechanisms, also a dose-dependent reduction of transcription was observable. In the comparison between male and female cells, it would be of interest to see whether the xenobiotic response elements at the core of the molecular mechanism actually recruit different proteins. To this end, CHIP assays were performed. In the male-derived cells, the recruitment of aryl hydrocarbon receptor was observed. This is the transcription factor that leads to the activation of the UGT1A genes that we studied. In the female cells, however, estrogen receptor alpha 1, in addition to histone deacetylases 1 and 2, was recruited, which leads to a chromatin remodeling and to a reduction of transcriptional activation or its absence. So this experiment shows that HDCA1 and 2, in addition to estrogen receptor alpha, are responsible for the difference between male and female. In the end, we took the in vitro experiments into the animal model. In animals, coffee treatment induces UGT1A expression. In females, this was not very effective in the colon and in the jejunum. By treatment with tamoxifen, we were able to restore this activity to normal levels, which corroborates the findings we saw in the cell culture that even in a living animal, the dependency on estrogen receptor alpha 1 is evidently at the core of the observed phenomenon. It was also interesting when looking at different tissues from these animals by immunofluorescence that in the organs that showed a very low UGT1A expression in females, the expression of estrogen receptor alpha 1 was very high. So in summary, there is a clear gender-specific regulation of UGT1A genes, a major detoxification enzyme family, and this is based upon aryl hydrocarbon receptor signaling, which is modulated by estrogen receptor 1 alpha and histone deacetylases 1 and 2. So what is the meaning in a more broader context of our study? This study indicates that there is a clear difference of metabolism between males and females. We can speculate that by similar mechanisms, also controlled by aryl hydrocarbon receptor or possibly by NRF2, these mechanisms may apply to more enzyme systems than the UGT1A genes. It is also possible to speculate whether transporter proteins underlie similar regulatory features. This is interesting because some of the genetic diseases related to variants in transporter proteins occur more often in female individuals. At least the clinical phenotype is more often observable. We feel that this may have broader implication for future pharmacotherapy and also for the view on diseases between males and females. It can be speculated whether a therapy one fits all is really what we should be doing, or whether more and more knowledge will be developed that differences in gender specificity regarding drug metabolism should be taken into account to adjust doses and to use the right medication for the right patient. This simple example shows two things. Drug metabolism is different, and possibly xenobiotic detoxification and the protection from geno and cytotoxicity can be different and is differentially regulated between men and women.